Welcome children. We are going to study today DNA replication, and in the previous videos we have seen that why DNA is preferred as a genetic material over RNA. Since RNA is not structurally stable and is not competent enough to be the genetic material. Here Watson and Crick, after presenting their model of DNA, where it was a double helix structure and the complementary strands were present to carry the genetic information. Watson and Crick also proposed at the same time the semi-conservative model for the DNA replication process. Now they proposed the semi-conservative mode of replication of DNA in 1953, and the highlights of the model were that the two strands of DNA which were complementary to each other, that means adenine was pairing with thymine and the guanine was pairing with cytosine, the nitrogenous bases. So the two strands which are complementary to each other would separate. That was the first step. The second step said that each strand would act as a template for the synthesis of the complementary strand. So out of the two strands which are present over there, the complementary strands, this strand will act as a template, and on the basis of this template, a complementary strand would be synthesized. So this would be the complementary strand. This was the second point which they suggested. The third point, they said, after the completion of the replication, each DNA would have the parental, that is the template, and one newly synthesized strand, that is the complementary strand, resulting into the formation or the passing of the genetic information. This was the model which was proposed by Watson and Crick. Now, the experimental proof for this mode of replication, that is the semi-conservative mode of replication, was given by Matthew Michelson and Franklin Stahl in 1958. They came up with an experiment to prove that the DNA replication happens semi-conservatively. And why we are calling it as a semi-conservative? We are calling it as a semi-conservative mode of replication because one of the parental strand remains intact and the complementary strand is formed on the basis of the template. So it is half of the information is retained and half of the information is generated on the basis of the parental strand. That's why we are calling it as a semi-conservative model of DNA replication. Now let us study what experiment was performed. Now the scientist took or performed the experiment on E. coli bacteria because of its fast multiplication. E. coli multiplies after every 20 minutes. So here E. coli was used for, that is the bacteria, Escherichia coli. It was used for studying the pattern of DNA replication. Now here E. coli was grown in a medium in a medium containing N95, that is a heavy isotope of nitrogen, and nitrogen was used because DNA consists of nitrogenous bases. So the nitrogen was introduced into the DNA, and for several generations the multiplication was continued. As a result, the DNA of E. coli was labeled with N15, heavy isotope of DNA. Also, it is not a radioactive isotope, but it can be separated out from the normal N14 of nitrogen of DNA by the density gradient. So we can differentiate between the N14 and N95, N14 and N15 DNA. Now, after growing the E. coli for several generations in N15 medium, it was completely labeled with heavy isotope of nitrogen. After 20 minutes, when E. coli divided, the density pattern was studied, and pattern was studied on the using cesium chloride as the density gradient. Now, cesium chloride is utilized because it clearly demarcates, and the density, the separation of the densities take place after the centrifugation. So after 20 minutes, the first generation showed that all hybrid E. coli, that is the DNA, one heavy and one lighter, that is one N15 and N14, were present. So both the strands, that is, it is a depiction only. So here all the DNA was having hybrid formation, that is one light strand and one heavy strand, or one hybrid, that is two hybrids were there. Now in the second generation, it was observed that 50% of the generation was hybrid, and the other 50 was the lighter generation. So this was 50% light. The third generation, after again the 20 minutes, it was observed that the third generation consists of 25% of hybrid and 75% light. Further allowing it to multiply for 80 minutes, the fourth generation had 12.5% of hybrid and 87.5% light. So here, after studying the pattern of DNA replication and allowing it the study through the density gradient using cesium chloride as a medium or the salt solution, it was found that definitely the replication in DNA is semi-conservative. Later on, studies were also conducted on Visia feba by scientists, and the medium, or that is the density gradient which was used over there, was thymidine, and it was found by Taylor and his co-workers that semi-conservative mode of replication is being followed by the DNA, and it was therefore established that DNA replicates semi-conservative method, that was proposed initially by Watson and Crick.